Dysregulated phosphorylation of pyruvate kinase M2 resulting from insulin resistance. A presentation by Austin Lorenz, an undergraduate student at Colorado Mesa University. Insulin is a hormone that is secreted by the pancreas when blood glucose is too high. As blood glucose rises, insulin becomes secreted and binds to receptors on cells, binds specifically to insulin receptors on cells that then activate a cascade of proteins downstream from the receptor that will then go and activate or inhibit proteins in different metabolic pathways in order to achieve needs that are useful for the cell. Specifically, protein pyruvate kinase M2 is a protein that catalyzes the reaction of phospholinopyruvate to pyruvate as the final glycolytic step of glycolysis. This step is important as pyruvate can be used to form ATP. It can be used for nucleic acid biosynthesis or fatty acid synthesis. Insulin resistance, however, is the inability of the receptor to recognize the binding of insulin when it is secreted from the pancreas. This results in the proteins downstream, the cascade of proteins downstream not being activated, which therefore do not go and regulate the proteins within the different metabolic pathways. For instance, when insulin binds to the receptor and does not activate the downstream proteins, pyruvate kinase M2 continues to be phosphorylated, which therefore deactivates it, which does not allow for the conversion of phospholinopyruvate to pyruvate. As this phosphorylation becomes dysregulated, there are intermediates that begin to build up and can lead to detrimental effects on the body. Some precursors for insulin resistance are as follows, as seen on this chart. One large correlation is obesity, as seen in the body metric stat. As obesity rises, there is an increased chance of insulin resistance forming. The general mechanism for which pyruvate kinase works, as I've kind of mentioned earlier, was that it becomes dephosphorylated, which then activates it. This phosphorylation is the primary effector or activator or inhibitor that leads to it performing its glycolytic functions. Specifically, the phosphorylation at tyrosine 105 is the exact location at which tyrosine becomes, or at which pyruvate kinase M2 becomes activated. As you can see in this graph, right here, Y105, as it becomes dephosphorylated, it becomes active, which allows for the conversion of phospholinopyruvate to pyruvate. Also, the upstream activators lead to this conversion. The specific protein is protein tyrosine phosphatase 1B, which is able to regulate this phosphorylation event. The protein tyrosine phosphatase 1B is the specific enzyme that goes and phosphorylates at this site. As you can see here, when dephosphorylated, you're allowed, or the glycolysis is allowed to go through and become final, and you can see the different sites of pyruvate going from pyruvate to lactate, in which you can produce ATP and use NADH. Pyruvate can also go into the citric acid cycle, in which can then go to the electron transport chain, creating large amounts of ATP for muscles. Also seen in this diagram is a lot of non-glycolytic functions that were unable to be discussed in this mini review due to restraints. Another type of phosphorylation that was important in regulating pyruvate kinase M2 was threonine 328 phosphorylation. This is another phosphorylation that is important in maintaining the stability of pyruvate kinase M2. Specifically, instead of protein tyrosine phosphatase 1B being the enzyme, it was heat shock protein 90 forming a complex with PKM2 along with another enzyme called protein kinase glycogen kinase 3 beta. These formed a complex together in phosphorylating threonine 328 on the PKM2, which therefore also activated the glycolytic functions of pyruvate kinase M2. The difference in this phosphorylation is that the phosphorylation leads to the activation, whereas with tyrosine 105 phosphorylation, it led to the inhibition of pyruvate kinase M2 from being activated and performing its glycolytic functions. Insulin can help lead to this threonine phosphorylation as it binds to the insulin receptors, communicating to the cell that there is a high blood glucose level, which needs to be taken up from the blood and converted into pyruvate, which can then lead to other beneficial effects of it. Some of the effects of insulin resistance on the body as this process becomes less regulated and the insulin is unable to signal to the cell that this glucose uptake needs to occur is one major one is inflammation. So as you can see in this chart, inflammation occurs in the systemic circuit as well as in adipose tissue, which then leads to insulin resistance. And then insulin resistance can also lead to free fatty acids, which will end up building ectopic fat. This is why you can also see the correlation between obesity and insulin resistance. As this insulin resistance occurs, there is a buildup of fat, which leads to obesity. There's also some research showing that these free fatty acids are the cause of pancreatic beta cell dysfunction. There is some treatment that is available for insulin resistance. The main treatment, there's not so much a pill you can take. However, it is a more physiological approach that you can take is through exercise. As you can see in this chart, as people become more active, the insulin sensitivity increases while insulin secretion decreases. This is important so that the body does not have to secrete so much insulin in order to achieve the same effect of getting glucose out of the blood as it would be necessary if the receptors are not sensitive to it. It has to pump out more insulin in order to get this same effect. As you can see here, as insulin sensitivity decreases and secretion increases, as sensitivity decreases but secretion stays the same, that's when you start getting into diseases such as type 2 diabetes and prediabetes, also seen here. It's not just exercise that is the regulator of insulin secretion, it's the type of exercise. It's not just getting up for 5 minutes, doing a few push-ups and saying, oh, I exercised for today, I'm not going to be a diabetic. As you can see in this chart, the time and duration of your exercise as well as the um, as well as how vigorous it is and how difficult it is, uh, changes insulin uptake and glucose uptake, or insulin secretion and glucose uptake. So up in this corner, you can see in the first 30 minutes, uh, glucose is rising in the blood as the body needs to do this, obviously, so that it can keep producing ATP in muscle cells. Also seen is the rise in insulin, which is a direct result of the glucose levels being increased. And then down here, you can see free fatty acids decreasing as um, as insulin secretion goes up. Another function of insulin is to stop the free fatty acids from, or I'm sorry, is to stop lipolysis, which is the breakdown into free fatty acids. As insulin secretion is increasing, it's not just that the secretion is increasing, you can tell that the sensitivity is also increasing as these free fatty acids are going down. Since they're going down, you can tell that insulin is performing its other function of stopping the breakdown of free fatty acids.